back to life. Imagine hearing a sound that hasn't existed on Earth for 10,000 years. A deep howl echoing across a frozen valley. A sound that disappeared long before the pyramids were built, before cities, before recorded history. For the first time in human history, we might be able to bring back traits of animals that went extinct thousands of years ago. Not through magic, not through cloning dinosaurs like in Jurassic Park, but through something much stranger, rewriting the DNA of living animals using the genetic code of the dead. One of the species often mentioned in this conversation is the direwolf, a massive predator that disappeared roughly 10,000 years ago, at the end of the last ice age. It looked like a wolf, but bigger, heavier, and built for cheese. Once the last individual died, that was it. The genetic instructions that defined that animal, millions or billions of letters of DNA, slowly broke apart over time. All that remained were bones, fossils, and sometimes fragments of genetic material trapped in ice or sediment. But in the last 20 years, scientists have developed tools that allow them to do something extraordinary. They can read ancient DNA and not dot small pieces. In some cases, researchers can reconstruct nearly complete genomes from animals that died thousands, sometimes even tens of thousands of years ago. One of the most famous examples is the woolly mammoth. Frozen mammoth remains discovered in Arctic permafrost are sometimes so well preserved that scientists can recover large amounts of genetic material from them. Well, to give you an idea of the scale, a mammoth genome contains roughly 4 billion DNA letters. That's about the same amount of genetic information as humans. Reconstructing that genome is like trying to assemble a shredded encyclopedia, except the pages are frozen inside bones buried in ice for 10,000 years. But reading the DNA is only the beginning. The real challenge is turning that information into a living organism. And that's where modern biotechnology enters the story. Instead of trying to clone a mammoth directly, which isn't possible with damaged ancient DNA, scientists use a different strategy. They compare the genome of the extinct animal with the genome of its closest living relative. In the case of mammoths, that relative is the ancient elephant. The two species are surprisingly similar. Their DNA is more than 99% identical. That means the genetic differences responsible for mammoth traits, thick fur, cold-resistant blood, fat layers, are hidden inside a relatively small number of genes. Scientists can identify those genes, and with gene editing tools like CRISPR, they can insert some of those traits into elephant DNA. The result wouldn't be a perfect mammoth. Instead, it would be something closer to a mammoth-like elephant, an animal carrying some of the key biological adaptations of the extinct species. According to the de-extinction process described by researchers working on mammoth restoration, the workflow follows several steps. First, scientists collect mammoth DNA from frozen remains found in places like Siberia and Alaska. Next, they sequence multiple mammoth genomes to understand which genes controlled cold adaptation traits. Then those genes are edited into elephant cells using CRISPR. If those modified cells develop successfully into embryos, they could eventually grow inside elephant surrogates, or possibly artificial womb systems being developed today. If everything works, the result could be animals capable of surviving in environments where mammoths once lived. But why attempt something this complicated in the first place? Part of the answer lies in climate science. During the Ice Age, mammoths were not just large animals wandering the tundra. They were ecosystem engineers. Imagine millions of giant herbivores moving across northern Eurasia. Each mammoth could weigh 6 tons, roughly the same as a large truck. As they moved, they knocked down trees, trampled snow, and grazed on vegetation. Over thousands of years, this constant activity helped maintain vast grasslands known as the Mammoth Steppe. At its peak, this ecosystem stretched across an area larger than the modern United States. And it may have played an important role in regulating the Arctic climate. Today, Arctic permafrost stores an enormous amount of carbon, around 1,600 billion metric tons. That's roughly twice as much carbon as currently exists in Earth's atmosphere. If that frozen ground melts, massive amounts of greenhouse gases could be released. Some scientists believe large grazing animals might help slow that process. By trampling snow and promoting grasslands, they could help keep the ground colder. In other words, create an extinct species. It might help stabilize entire Arctic ecosystems, at least in theory. Of course, not everyone agrees this is a good idea.
Introducing genetically engineered animals into modern ecosystems raises a lot of questions. For example, what the ecosystem can support, or if they spread diseases to existing wildlife. Even small ecological changes can have cascading effects. For example, when wolves were reintroduced into Yellowstone National Park in the 1990s, the entire ecosystem changed, from deer populations to river patterns. Now imagine introducing animals that haven't existed for 10,000 years. The ecological consequences could be unpredictable. There is also another concern scientists often discuss. It's sometimes called the moral hazard of de-extinction. The idea is simple. If people start believing that extinct species can always be brought back later, they might feel less urgency to protect endangered species today. Why worry about extinction if science can reverse it? However, studies examining public attitudes suggest the situation may be more complicated. Simply knowing about de-extinction doesn't necessarily make people more comfortable with species disappearing. But scientists still emphasize something important. De-extinction cannot truly reverse extinction. Even with advanced gene editing, recreating an identical species is extremely unlikely. What scientists can create are genetic approximations, animals that resemble extinct species and may play similar ecological roles. But they are not perfect copies of the past. And yet, the technologies developed for de-extinction may still have enormous value. The same genetic tools could help scientists protect endangered animals today. For example, gene editing could potentially increase genetic diversity in small populations, or help species adapt to changing climates. In other words, the real legacy of de-extinction might not be resurrecting lost species. It might be preventing future extinctions. For thousands of years, extinction seemed like a one-way door. Once a species passed through it, there was no way back. But modern biotechnology